Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 26. The topic is processing of mRNA. Discussing agenda for the topic will be eukaryotic cells modify RNA after transcription, split genes and RNA splicing. Lecture outcome. After watching and listening this lecture, students will know about mechanism involved in modifying the primary mRNA transcript. Enzymes in the eukaryotic nucleus modify pre-mRNA in specific ways before the genetic message is dispatched to the cytoplasm. Eukaryotic genome has non-coding regions, and during the process of transcription, they are also copied to the messenger RNA. So these non-coding regions must be removed. And secondly, when messenger RNA enters cytoplasm, enzymes takes on it and starts degrading it. So there must be some kind of protective mechanism to take care of messenger RNA in the cytoplasm and prevents its premature degradation. During this RNA processing, both ends of the primary transcript are altered, means the addition of 7-methyl GTP head and 3-polyatyl. Also, in most cases, certain interior sections of RNA molecule are cut out and the remaining parts spliced together. Here they are talking about splicing, removal of introns, and alternative splicing, the rearrangement of exons. The modification produce an mRNA molecule ready for translation. So once the process of transcription is completed, the RNA product which is formed is called pre-mRNA, and during the post-transcriptional modification, the RNA becomes mature and ready for translation. Now here you can see uh, RNA processing. Here is the addition of polyatyl, means 50 to 200 adenine nucleotides are added to the 3' end, and 7-methyl GTP head is added to the 5' region, and furthermore the introns or the non-coding regions, if present, are removed, and the exons or the coding areas are brought closer to each other. Alteration of mRNA ends. Each end of a pre-mRNA molecule is modified in a particular way. The 5' end, which is synthesized first, receives a 5' cap, a modified form of guanine nucleotide added onto the 5' end after transcription of the first 20 to 40 nucleotides. The 3' end of the pre-mRNA molecule is also modified before mRNA exits the nucleus. So the addition of cape and poly-A tail, it increases the longevity of messenger RNA and furthermore is helpful in the settlement of messenger RNA at a correct orientation, at a correct position on ribosome before translation. At the 3' end, an enzyme then aids. So here we have already discussed polyadenylation in transcription, where polyadenylation is a particular sequence consists of those nucleotides, and when this particular sequence arrives in the gene, it signifies the fact that the gene or the process of transcription will end anywhere between 10 to 25 upcoming bases. So it is actually a signal for termination, and it is also copied on messenger RNA. And once it is copied and messenger RNA is released, then further 50 to 250 adenine nucleotides are attached with that sequence in the messenger RNA. The 5' cape and poly-A tail share several important functions. First, they seem to facilitate the export of mature mRNA from the nucleus. Second, they help protect the mRNA from degradation by hydrolytic enzymes. And third, they help ribosome attach to the 5' end of mRNA once the mRNA reaches the cytoplasm just before translation. The UTRs in the previous figure will not be translated. So this polyadenylation signal and the poly-A tail and the nucleotides which are added to the 5' cape, these are the UTR regions. They are referred as 5' and 3' UTRs. UTRs means untranslated regions. They will not be translated, but they are there only to perform those three functions. Split genes and RNA splicing. Now, as already discussed, the eukaryotic genome is enormous because of the presence of non-coding regions or introns, and these non-coding regions does not represent any amino acid, and these triplets must be removed before the process of translation starts. A remarkable stage of messenger RNA processing in the eukaryotic nucleus is RNA splicing, where large portions of the RNA molecules are removed and the remaining portions are reconnected. The cut and paste job is similar to editing a movie. Means there are certain regions which are not beneficial introns, which does not represent amino acids, so they must be removed, and the coding regions which represent amino acids must be brought closer to each other. This whole process is called splicing. The average length of a transcription unit along a human DNA molecule is about 27,000 nucleotide pairs. So the primary transcript is also that long. Means it is the average length of a transcription unit. Transcription unit means the area of the gene which is copied on RNA transcript. So if the transcriptional unit or the area which is transcribed is 27,000 nucleotide pairs, so messenger RNA will also be 27,000 nucleotides long. But the average size of proteins is 400 amino acids. This is an average-sized protein transcribed by a gene. And you know that one amino acid is represented by three nucleotides. So 400 into 3 makes 1200. So it means that out of those 27,000 nucleotide pairs, only 1200 nucleotides encompasses the exons or the expressed regions. The rest of the nucleotides are the introns or the intervening sequences that needs to be removed from the messenger RNA before translation. This is because most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides that are not translated. Even more surprising is that most of these non-coding sequences are interspaced between coding segments of the gene and thus between coding segments of the pre-mRNA, means the coding and non-coding regions are present, they are intermixed. There are suppose 200 to 300 intron sequences, then after that there is some sequences which represents amino acids, then again there are certain sequences which are intronic. In other words, the sequence of DNA nucleotides that codes for a eukaryotic polypeptide is usually not continuous. It is split into segments, means the coding regions and non-coding, then the coding regions. So these non-coding regions are called intervening sequences or introns. Here you can see messenger RNA 5-cap 3-polyatyl. This is the exon or the expressed region. This is also an exon, and this is also an exon. And this long sequence is the intron, the non-coding sequence. The triplet of bases located here does not represent any amino acid. So therefore, this particular region must be removed from it. And this region is cut out and the coding regions are joined with each other. So this intron will be removed and this one will be removed, and the coding regions will be rejoined with each other. So this is the actual area which represents the information for the synthesis of a polypeptide chain, and this area is 5-UTR, this is 3-UTR. You already know its function, they will not be translated. The area only in the red shade will be translated. So exons are eventually expressed, usually by being translated into amino acid sequence. Exceptions include the UTRs of exons at the ends of the RNA, which make a part of the mRNA but are not translated into proteins. Because of these exceptions, you may prefer to think of exons as sequences of RNA that exit the nucleus. So only that particular RNA will leave which only has exons and all the introns have been removed. Because introns and exons both are present in the DNA, therefore they are copied to the RNA, and then before translation they are removed. The non-coding areas are removed from the RNA. Now we will confine our attention slightly to the splicing process, how the process of splicing takes place, how the uh, enzymes responsible for this process carry out their function. So the removal of introns is accomplished by a large complex made of proteins and small RNAs called a spliceosome. Spliceosome is a large complex of several different types of proteins which forms which carries out this function. These small proteins are also in association with snRNAs. So these snRNAs are quite often abbreviated as SNRPs, small or ribonucleoproteins. Here you can see this is exon coding region. This one is also coding region. This is the long non-coding intron. So a spliceosome complex comes, this spliceosome complex has small RNAs and these small RNAs along with other enzymes cleaves those ends and detach the introns from the coding regions, and the coding regions are rejoined with each other. And how the spliceosome recognize that this particular sequence is an intron and it needs to be removed. There is a GUAG rule. GUAG rule means that an intron will always have GU nucleotides on one side and AG nucleotides on the other extreme. So these spliceosomes, it is a signal that the sequences residing between GU and AG are non-coding and needs to be removed. So the introns is then released and rapidly degraded, and the spliceosome joins together the two exons that flanked the introns. It turns out that the small RNAs in the spliceosome not only participate in spliceosome assembly and splice site recognition, GUAG is the splice site, but also catalyze the splicing reaction. It is the end of the lecture. Thank you very much. Allah Hafiz.